Hello there, and welcome back to Hydroponics Daily, your go-to podcast for everything soilless cultivation. I'm your host, Dr. Russell Sharp, the founder of Utrema Limited, where we make interesting fertilizers, novel biostimulants, and organic biopesticides. However, before I was an entrepreneur making fertilizers and inventing new fertilizers and things like this, I was an academic plant scientist, a plant physiologist to be precise. And one of the things you do as a plant physiologist, certainly where in the area I was working in, was study water deficits. And key to that is looking at stomata. Now, stomata are the pores on your leaves that allow gases to be exchanged with the air. Simple as that. Now, stomata are the holes. They are not the structure. So they're just the hole, just like the pupil of your eye. Although I am not an animal scientist, so I don't even know that it's true or not. So basically, a stomata is made up of two guard cells. And those guard cells are very clever. They can fill with liquid and close, or they can empty of liquid and open. Okay? So it depends on their turgor pressure, how open or closed the stomata are. And when the stomata are closed, the plant can limit the amount of water that it loses. And that's useful if they become stressed. If there's a drought, then they can close their stomata and limit the amount of water they lose. The same at night. They don't need to open their stomata at night, so they close them and limit the water they lose. Apart from tomatoes, apparently. Sorry, potatoes. Potatoes, apparently they keep their stomata open 24 hours a day. Probably because they're growing in. They need the calcium to get, and the calcium is absorbed in the soil, so they just keep them open all night, get that calcium in, and they're growing in soil that's got loads of water anyway, so they don't bother closing them at night. That's what I'm told anyway. I've never actually researched it or looked at it, but that's what I'm told from the research. Now, why even open them in the first place? So the reason why they open them and they lose water is because they need to exchange gases. CO2 needs to diffuse in. It doesn't suck in, it has to diffuse in, and oxygen has to diffuse out. And a byproduct of that is that water escapes at the same time. Just like when you're breathing, you lose a lot of water in your breath. You can't help that. If you're in the desert, you can't help losing water in your breath. So a plant can't help losing water when it's exchanging gases. The inside of it, the leaf, is covered in liquid. All the cells are wet inside a leaf. And that's because it aids CO2 and O2 diffusion back and forth into the air. Just like your lungs are all wet. If you were to go inside your lungs, it would be covered in moisture, and that's to aid oxygen absorption and CO2 release. So the same thing in your lungs as happens in a plant, except the different gases are going the different way. Obviously, in your lungs, you breathe in, you suck in, but plants don't do that. So they have to rely on diffusion. So the leaves the cells inside the leaf have to be wet for that exchange to happen. So there's a bit of a balancing act between the amount of water lost and the amount of CO2 that's being absorbed. Now, things like cacti, we've talked in the past, they can absorb CO2 at night. So they open their stomata at night and then close them during the day, and that limits the amount of water that's being lost. But most plants, they're open them during the day, and the peak happens around 12 o'clock, noon, midday, which in the summer is at 1 o'clock. And the stomata regulates that. Very little diffusion occurs through anywhere else in the plant, gas exchange. And most of the stomata are positioned on the underside of the leaf. So 95% of all the stomata are on the underside of the leaf. Most plants have next to no stomata on the top side. The top side is normally waxy. And you take a house plant and look at it, you'll find the top is very waxy and a different colour to the underside. That's because that's where the gas is exchanged on the underside, and the light is being absorbed at the top. So if you look at the structure of a leaf, you'll find what's called the palisade cells. It's called the palisade cells because they look like a fence, the striped fence. The cells are all lined up. Their job is to capture sunlight, and they're at the top of the leaf. And the ones at the bottom, they're spongy. They look like the lungs, the cells in your lungs, and they are there to exchange gases. That's their job. Now, some plants that will have equal stomata on both sides, so maybe like certain grasses or things like that, but they are the exception and not the rule. So we said the stomata are the holes, and the aperture, how wide they're open, governs how much they'll lose. The amount of stomata can be an influence on how much gas exchange occurs, but it's really about how open they are, because the cells push together, the guard cells, and tightly close and limit virtually any water loss. Now, how open they are and how close they are, and how much water gets lost from the stomata is measured using something called a pyrometer, and that measures just got called away there. Where was I? I was measuring the stomatal conductance, how easily gases are exchanged, how open the stomata are, and that's measured using a pyrometer, as I said, and it's measured in the units micromoles per meter squared per second. So how much gas can get through a square meter per second. And using a pyrometer is really quick and easy. It's a lot less laborious than putting leaves under microscopes and seeing how open they are and measuring how open the aperture is. And also, you've got to remember that as soon as you take a leaf and put it under a microscope, you're changing the conditions. So we might get a lot more stressed and not really being representative. So you can use stomatal conductance as a measure of stress, a measure of water stress particularly. And so when you dry a plant out, or even when you flood it, it will close its stomata. If you stress it in any way, it'll probably close its stomata. So pyrometers are really useful pieces of kit, and they've been used in plant physiology for a while for measuring how open the stomata are and how much gas is being exchanged. Unfortunately, they're still really expensive and not widely available, and so you can't really integrate them into a medium or small scale hydroponic unit. You might have one to experiment with if you're a large scale hydroponic grower, but pyrometers still are out of the realms of most growers, unfortunately, because the technology is not really epic. It's just nobody's decided to produce a low cost option because not many people want them. So unfortunately, that's the case. However, there is an easy way of looking at your stomata and seeing if they're open or closed. And it's not to just put your leaf under a microscope, because if you do that, I've told you in the past about how dissecting microscopes are really good. You just throw a leaf or throw a mite or a spider mite or an insect under there and you can look at it. Whereas the normal microscopes, the type you could talk about using in school or university, they're compound microscopes. And for them, you need a thin layer to actually see anything, because the light needs to travel through the cells. And so you have to use something called a microtome to make small slithers and then put them on a microscope slide and put some water there and a cover slip and be very careful. So that's not really, you can't really do that with stomata, because by the time you've done that, the stomata have all closed. What you can do, and this is my top tip, and definitely you should try this because you can get some wicked images doing it, is to take your leaf on your plant and then apply a very small layer of clear nail varnish to it. So when I was a plant physiologist, I had a drawer full of clear nail varnish, even though my nails are terrible. And I used to paint on nail varnish onto my leaves. And then after it's dried, you peel off the nail varnish very carefully. Otherwise, you can deform the image of the cells that you get. And you get an absolute mirror image of your cells and anything else that's on the leaf surface. So if it's trichomes or unusual shaped epidermal cells, veins, xylem, phloem veins, then you can picture all this and you get a black and white image of what it looks like on your leaf. So you take the peel, you put it on a microscope slide, place it nice and flat, maybe we'll put a cover slip over it, and then just look at it under a microscope, times 10, times 40, and you can see all the stomata, and you can see if they're open or closed. You can see how many there are, the density, and in plant physiology, we use this method, the nail varnish technique, mainly to look at density, to count how many stomata there are per square millimeter. And that's really useful because if you stress the plant for long enough, the new leaves that produced may have more or less stomata, more or fewer stomata. And so that's a really good, useful tool. So although it's a easy and almost a silly method, it is very useful. It's one of the methods we use to study stomata. So I definitely recommend that. And if you're just curious about looking at your stomata, it's really good. So hydroponic growers tend to be very inquisitive, tend to be into botany and plant science. And so even if you have no need to measure your stomata and look at your stomata, you'll probably do this now. You'll get some clear nail varnish from Boots or Superdrug, or don't know, where do you get nail varnish in America? CVS, something like that. Or from your girlfriend or your partner or your wife, or your man, or yourself, or your mum, or your sister, or your daughter. And you'll be applying it to your plants, all your different plants that you're growing, your house plants, your crops, and seeing what the stomata look like. If you find anything useful, interesting, then please do let me know. Send me a message. If you've got other ways of measuring stomata, then let me know as well. If you found this podcast useful, then please leave us an Apple podcast review if you're listening on Apple. If you're not listening on Apple, leaving a review is useless. So please just check out utrema.co.uk. And if you're struggling to spell utrema, it's Latin for wasabi. So just Google that. Thank you very much for listening to Hydroponics Daily for today, and I'll see you again tomorrow.